Hi everyone, myself Dr. Girisha, working as the Assistant Professor in School of Agriculture, SR University. So today in this session, we'll discuss about diseases of mango and their management. So here you can see the list of important fungal, bacterial and viral diseases in mango. Anthracnose of mango, which is caused by Cultivation gliosporides. Then powdery mildew of mango, which is caused by Idium mangifera. Then mango malformation, which is caused by Fusarium subglutinans. Then stem and rot in mango, which is caused by Lacidiplodia theobrome. Then sooty mildew of mango, which is caused by Capnodium bromazum. Then dieback disease in mango, which is caused by Certocystis fimbriata. Then Phoma blight of mango, which is caused by Phoma glomerata. Then rust disease in mango, which is caused by Cephalovirus virescens. Then bacterial canker or bacterial blight of mango, which is caused by Pseudomonas campestris, Pethor mangifera indica. Then finally, pink disease in mango, which is caused by Pellicularia salmonicola species. So these are the list of important fungal and bacterial disease in mango. First, let's start with the anthracnose disease of mango. So this anthracnose disease of mango, which is caused by Coltotrichum gliosporides. So coming to the major symptom of anthracnose of mango, first the fungus produces leaf spot, blossom blight, wither tip, twig blight and fruit rot symptom. On the leaves initially circular light yellow brown spots are formed, then which soon enlarge with a dark brown to black margin. The disease spreads rapidly during rainy season and covers the tender twigs, floral parts, including the floral axis, and causing them to turn black and they will become dry and which will most similar like a dieback symptom appear on the inflorescence. If the infection is severe, individual spots coalesce leading to twisting of the foliage and premature defoliation will occur. Then falling of the entire leaves. On the fruit spot appear near the stem and small brown area that enlarge rapidly and they will become dark brown to black in color. In some cases, in the areas involved, they form the streak like a parallel streak formation occurs on the fruit. Fruit fall beneath the spot, it will become very hard and you can see the cracking and decay at the ripening stage. Infected fruit may also drop prematurely. The latent infection is carried from the field and develops further. When we store the mango in the storage condition, there is a development of the symptom. So this is example for latent type of infection because infection is already there from the field condition, but the expression of the symptom occurs during storage due to favorable condition. Generally, all the varieties of mango are mostly susceptible to anthracnose disease. These Neelam and Bangalore varieties are highly susceptible to mango anthracnose. These are the pictures representing the anthracnose disease in mango. You can see the complete inflorescence which is infected by anthracnose pathogen Coltotrichum gliosporides. Here also both the leaves and inflorescence that flower is infected with the mango anthracnose pathogen. So here we can see like the end of the stem end which is infected with the mango anthracnose pathogen, and we can see the streak or line formation occurs on the fruit, and there is a symptom development occurs during storage condition, and also dark brown to black color spot formation occurs on the like young and immature fruits. So overall these are the pictures representing the anthracnose disease of mango. Then you can see under storage condition there is a development of large dark brown to black color spot formation occurs on the fruit, which will greatly reduce the market value, and these affected fruits are mostly unfit for consumption, and there is a great decrease in the sucrose content in the fruits. Coming to causal organism, this mango anthracnose, which is caused by Coltotrichum gliosporides, perfect stage of the fungus is Glomerilla cingulata. Coming to the mycelium of the fungus, which contains separate branched and slightly dark colored hypha. The hypha develop the fruiting body, which is known as acerulais or acerulus. The acerulus produces closely packed single-celled colorless conidiospore that produces oval to oblong conidia on their tip. The conidia are non-septate, measuring about 12 to 16 4 to 6 micrometer in diameter, and sometimes possess oil globules in the acerulae. These acerulae produce sterile air-like structure within the fruiting body, which is known as setae. So this is the image representing both the conidia and sterile air-like structure setae along with the fruiting body acerulae. Coming to the disease cycle of mango anthracnose, majorly coming to primary infection, mainly through previous season infected leaves, like infected plant debris, defoliated branches, or mummified flowers and fruits, they will act as a primary source of infection. Coming to secondary spreading, mainly it is through conidia produced on the acerulae, which will be carried by the rain or wind or rain splashes. Coming to favorable condition, it requires a temperature of 25 degrees Celsius with a RH of 95 to 97%. Moist and humid condition are highly favorable for the disease infection. Then coming to management of mango anthracnose, we need to avoid overcrowding of the orchards, go for optimum spacing. Tree sanitation is very important. Proper training and pruning is highly recommended. We can remove the disease peaks at the time of pruning and training, and after training and pruning, we need to apply Borax paste to the plants to avoid any infection. Then application of carbendazole or topsin 0.1% or chlorothalonil 0.2% at 14 days interval until the harvest is highly recommended to control the anthracnose disease. So before storage, treat the fruits with hot water at the temperature of 50 to 55 degree for a duration of 15 minutes, or we can dip the fruits in a benomyl solution 500 ppm or thiobendazole solution 1000 ppm for 5 minutes to control this anthracnose disease during the storage condition. So this is regarding the symptoms, disease cycle, and integrated management of mango anthracnose. Then coming to malformation in mango, very important disease affecting mango growers. This mango malformation, which is caused by Fusarium moniliforme subglutinins. Coming to economic importance, this is one of the severe disease of mango important in northern India. In Andhra Pradesh, this was first noticed in Aragonda village in 1971. In India, it is known to occur in UP, Maharashtra, Haryana, Bihar, Punjab, Andhra Pradesh, Telangana, particularly severe in Uttar Pradesh, causing much damage. In Northwest India, nearly 50% incidents on orchards were recorded, whereas in Northeast India and South India, it may goes up to 10%. Coastal areas are usually free from the disease infection due to climatic factors. Coming to the symptoms of mango malformation, we are divided into two types, vegetative malformation and floral malformation. Coming to vegetative malformation, which is more pronounced in young seedling, and the seedlings trees less, which is less than trees than in grafted plants. The affected plants develop excessive vegetative branches which are of limited growth and a swollen tissue and have short internode. Sometimes the branches show bunchy top appearance, compact leaves are formed in a bunch at the apex of the shoot or in the leaf axil. The axillary buds of the affected branch usually swollen in case of older plants, but growth of the branch is not impaired. Coming to another type of malformation, which is floral malformation, mainly it will infect in the mango inflorescence or mango fruit. The flowering panicle, instead of coming out as a normal, it just appear as a compact bunch of odd flowers. Individual flower is greatly enlarged and has a large disc. The inflorescence get hypertrophied due to that hypertrophy and hyperplasia. Hypertrophy means increasing the cell size. Hyperplasia means increasing the number of cells. So due to this hypertrophy, there is a inflorescence will get swollen, and the percentage of bisexual flowers is malformed and the panicle is very low, and it will indirectly affect the fruit bearing capacity of mango tree. These are the pictures representing the floral malformation. You can see due to hyperplasia and hypertrophy, there is a swollen inflorescence and complete malformation of the inflorescence. Sometimes there is no fruit bearing, and the reduction in the number of bisexual flowers also occurs during malformation. Coming to vegetative malformation, short internodes, the plant will appear like a compact bunch of leaves, and a reduction in the plant height also occurs. Majorly, this vegetative malformation occurs during 2 to 3 years age of the mango plant. Coming to causal organism, Fusarium moniliforme, variety subglutinins, which is severe biotic and abiotic factors also reported to cause malformation such as physiological disorder, Eriophyid mite, and virus infection. It is now established that this pathogen, Fusarium moniliforme subglutinins, is a major causal organism involved in causing mango malformation. So this Physarum, it will produce the micro and macroconidia. Coming to microconidia, which are one or two celled oval to fusiform in shape. Coming to macroconidia, which are rarely produced, are there two to three celled and falcate in appearance. And also this Physarum will produce chlamydospore, which is a asexual structure which is able to survive under unfavorable condition. So this is the disease cycle representing the life cycle of Physarum moniliforme. First, airborne conidia, they carried by the wind and they will directly land on the new inflorescence or new bud and they will cause direct infection. Sometimes the mites also carry the pathogen indirect way in a passive manner. They will also infect the inflorescence. After that, there is a appearance of the symptom. Apical bud get colonized with the Physarum moniliforme and this might also infect the growing bud. Then there is the expression of the different kind of symptom, malformed inflorescence, malformed vegetative growth, symptomless branches which are already colonized with the Physarum mangiferae. Then aerial dissemination of the conidia of Physarum mangifera, mainly through wind or airborne conidia. So this is complete disease cycle of Physarum moniliforme. Then primary infection you already know through malformed inflorescence. Either secondary infection may be either through airborne conidia or the conidia carried by the Eriophytmite Escherichia mangifera. Coming to epidemiology, younger plants are more susceptible. Vegetative malformation reduces with growing age. Mild temperature of 8 to 27 degree and high RH are conducive for the disease infection. The incidence of mango malformation has been found to vary from season. Studies have been revealed that the temperature is closely related to the variation in the incidence of mango malformation. The seasonal variation in the temperature at the time of flowering was correlated with the intensity of malformation. This trend also reflected in the world distribution of disorder and its incidence is mainly recorded where the mean temperature during the winter is less than 16 degree. However, the temperature does not appear to have major role once the symptom have appeared on the panicle because of the affected panicle continue to grow, producing excessive growth in dry hot summer. So we can see this temperature as a close relation with the appearance of the disease. Then coming to management practices to control the mango malformation, it is advisable to avoid the season stick from the trees bearing malformed inflorescence for the propagation. Indexing of healthy mango tree to be done to serve as a material for grafting. Only certified saplings should be used for propagation. As soon as the disease symptom is expressed, the plant terminal bud should be pruned along with the continuous 15 to 20 centimeter apparently healthy portion and we need to burn the affected portion. A healthy orchard located in disease-prone pockets should be sprayed with a fungicide or carbendazole as a prophylactic application to avoid further occurrence of the disease. Then acaricide like propagates 57% EC is also recommended to control the mite infection. And also application of 200 ppm of NAA, naphthalene acidic acid, in the first week of October is recommended uh, to control the mango malformation, which is followed by we can go for deblossoming at the bud burst stage to control malformation. Then early flowering should be deblossomed. We can go for late flowering. So all these are IDM practices recommended to control malformation disease in mango. Then coming to another important disease in mango, which is powdery mildew disease in mango. Coming to its economic importance, the disease is worldwide in distribution, reported in India, Pakistan, Sri Lanka, and South Africa. In India, the disease particularly destructive in UP, Maharashtra, Karnataka, Andhra Pradesh, Tamil Nadu during the month of December to March. Coming to the major symptom of powdery mildew in mango, the disease can easily be recognized by whitish or grayish powdery growth on the inflorescence and tender leaves. Generally, infection starts from the inflorescence and spread downwards, covering the floral axis, tender leaves, and stem region. Leaves become twisted, curled, and they will become like malformed leaves, and finally, premature defoliation will occur. Infected floral parts are severely damaged and they will drop off. If the fruits are set, they do not grow in size, they may be dropped before attaining the peak size. Fruits are sometimes malformed and discolored due to severe mildew attack. Because of the fruit set, heavy flower and fruit drop, the loss due to disease may go as high up to 70 to 80%. So these are the images representing the whitish powdery growth formation on the affected inflorescence, then on the affected young immature fruit also. Then it will cause severe fruit dropping and severe defoliation. Coming to favorable condition, the disease is favored by warm, humid weather with cool nights. Causal organism is Idium mangifera. Sexual stage is Recipe polygony. Mycelium is branched, hyaline, superficial, septate, astoria, lobate. Conodia are hyaline, unicellular, elliptical, bond singularly are rarely in chains up to. Conidiospores are simple, erect with two or more basal cell. So this is regarding the etiology of the pathogen, Idium mangifera. Then coming to mode of spread and survival, dissemination by the wind and the progress of the disease in the orchids along with the direction of the wind, and primary infection mainly through infected plant debris. The pathogen survives in the form of dormant mycelium and also conidia. Secondary infection may be through conidia spread through wind. Coming to management, can be controlled with preventive spray with a wettable sulfur 0.3% once the before flowers open and the second after the fruit set. Dusting twice or thrice with the sulfur will check the disease incidence. Spraying with the keratin, or we can also go for application of any triazole fungicide. Some resistant varieties like Neelam, Bangalore, and Janardhan, person which are available to cultivate against powdery mildew disease in mango. Then coming to another disease, dieback disease, which is caused by Laccidiplodia theobrome. So coming to the major symptom, the pathogen causing dieback, tip dieback, Graftonian blight and twig blight and seedling rot, wooden stain, stem and rot, black root rot, fruit rot, dry rot, brown rot of panicle, etc. are the major symptom of this pathogen, Laccidiplodia theobrome. The disease is most conspicuous during the month of October to November. It is characterized by drying the back of the twigs from tip to downwards. Dieback means death of the branches from tip to downward, particularly in the older trees, followed by drying of the leaves, which gives an appearance of fire scorch. Internal browning in the woody tissue is observed when it is slit open along with the long axis. Crack appearance on the branches and gum exudes before the death of the plant. So all these are the major symptom of dieback disease in mango. Coming to the disease development, pathogen survives in the plant debris, which is in the source of primary inoculum. High humidity and moist conditions favors the disease development. The disease is most common during October and November month. These are the pictures representing the gummy secretion, development of cracks in the plants, and death of the twigs or branches from tip to downwards. So this is regarding disease development and major symptom of dieback disease. Coming to management of dieback, the cyan wood selected for propagation should be free from any kind of infection. Every case should be taken to prevent introduction of disease in newly planted orchards. Any infected portion should be immediately pruned, followed by spraying or pasting of copper oxychloride or pasting with the cow dung at the cut ends of the tree. Pruning should be done in such a way that some healthy portion is removed to ensure the complete eradication of the pathogen, like 3 inches below the infection site. And then coming to another disease, Diplodia stem and rot, which is caused by Diplodia natalensis. Coming to the major symptom, epicarp will become darkens around the base and pedicel region also. Circular black patch which under the humid condition. Soft rot head of pectinolytic and cellulolytic enzyme. More portion of the fruit turn which will turn to black and will become softened with a soft rot decay symptom, and the loss of ascorbic acid and non-reducing sugar rapidly occurs during infection with the Diplodia natalensis. Coming to disease development, the fungus persistent in the infected plant parts which severe is a source of inoculum with the RH of 80% with the temperature ranging from 25 to 31 degrees Celsius. So this is regarding Diplodia stem and rot. These are the images representing the stem and rot caused by Diplodia. Then coming to Poma blight or Poma glomerata, Coming to the major symptoms, symptoms of the disease are noticeable only in older leaves. Initially, the lesions are angular, minute, irregular, yellow to light brown, scattered over the leaf lamina. As the lesion enlarge, their color changes from brown to cinnamon and they will become almost irregular in shape. Fully developed spars are characterized by dark margin, dull gray necrotic center. In case of severe infection, such spars coalesce forming patches measuring 3.5 to 13 centimeter in length and resulting in the complete withering and defoliation of the infected plant. Coming to disease development, the pathogen is a seed-borne in nature. An inoculum present in the seed is a source of major primary inoculum. Fungus also survives in the glooms, fruit, and plant debris in the rainy season, which will majorly favors the disease development. Coming to management, we can follow common cultural, mechanical, biological practice and application of some systemic fungicide like carbendazim along with the mancozeb or chlorothalonil or any other systemic fungicide. Along with, we can also, after pruning or training, we can go for application of borax paste and borax mixture and balanced nutrition, which will provide resistance to Powdery blight. These are the images of Powdery blight. Then coming to physiological disorders of mango and their management. So coming to some important physiological disorders, which is first one is a black tip in mango. Coming to the major symptoms become visible when the mango fruit attain the marble size. Small etiolated area develop near the distal end of the fruit, which gradually spread and turn nearly black and cover the tip of the fruit completely. The black area remain hard and the growth of the fruit is checked black tip disease. So this is the symptom of black tip. Coming to management, internal necrosis, it can be minimized with the application of borax 1%. The first spray should be done positively at the P stage, followed by 2 more spray at 15 days interval. And also we can follow other cultural and mechanical methods to control the disease. Planting of mango orchard in north-south direction 5 to 6 kilometers away from brick kiln reduces the incidence of black tip disease in mango. So these are the images representing black tip disease or black tip physiological disorder in mango. Then coming to internal necrosis, majorly which is caused by boron deficiency. First, water-soaked grayish spot develop on the lower side of the fruit. Later, the spots enlarge and develop into dark brown necrotic area, and internal tissues start disintegrating, resulting in the exposing of flesh. Mango pulp. Yellow colored droplets also come out from the infected fruit. Coming to management, we can also go for foliar application with the borax 1% at the P stage of the fruit, followed by 2 application at 15 days interval. Application of 250 gram boron per tree at 10 to 15 days, 10 to 15-year-old plant around the tree basin. Then coming to another major physiological disorder in mango, which is spongy tissue in mango. This spongy tissue in fruit develops non-edible sore patch in the mesocarp of mango. It is in specific in Alpanso mango. Fruit from outside look normal, but when we cut it open, inside the patch of flesh become spongy, yellowish, and sore in taste. This can be detected only on cutting the fruit. Inactivation of ripening enzyme due to high temperature, convective heat, and post-harvest exposure to sunlight are the causes of spongy tissue in mango. Coming to management, use of mulching are useful reducing the incidence. The mango hybrids Ratna and Arka Punit, which have alphonse like character, do not suffer from this disorder. Harvesting the mangoes when they are 3 to 4 matured rather than fully matured also reduces the incidence of spongy tissue. The use of wind breaks for protecting the orchard from warm air during the May month, which can greatly reduce the incidence of spongy tissue. So these are the images. You can see the mango field internally affected with the spongy tissue disorder. Then coming to another important disease in mango, which is mango bacterial canker caused by Xanthomonas campestris Pethover mangifera indica. So these are the images representing different kind of symptom of mango bacterial canker. First, coming to the major symptom, the disease is found on the leaves, petiole, twig, branch, and also affecting the fruit. The disease fruit first appear as a minute water-soaked lesion, irregular lesion on any part of the leaf and leaf lamina. Several lesions they will merge together and it will give like necrotic canker patches on the infected leaf. In a severe infection, the leaves become yellow and there is a premature defoliation occurs. Cankery lesions also appears in the petiole region, twigs region and also young affected fruit. And the water soaked lesion also develops on the infected fruit, which later turn to dark brown to black in color. They often burst open and releasing high contagious gummy ooze containing the bacterial cell. Here the causal organism is Xanthomonas compactis, Pathover mangifera indica, which is a gram-negative bacteria. Coming to mode of spread and survival, infected nursery trees have been a major source of the disease in new orchard. Bacterium enters the leaf through stomata, in fruit and twig through lenticel. The bacterium survives in the infected parts in the tree. The pathogen survives up to 8 months in the leaves. The bacterium from canker and the twigs are the cause for primary infection source. Disease spread rapid during the rainy days. Disease spread to the new area through infected planting material. The fruits are found to in the bunches of disease spread. They can when they're coming in contact with each other. Coming to control measure, we can apply streptocycline 0.01% or agromycin 0.01% after first visual symptom appearance. Monthly spray with the carbendazim, bevestin or copper oxychloride, which is effective in controlling the diseases. Then coming to some important minor disease in mango. First one is sooty mold in mango, which is caused by Capnodium romazum. The sooty mold in mango tree appears as a dark, often black to velvety coating on the leaves, twigs and fruit caused by the fungi that grow on sugary secretion like honeydew secretion by the sap sucking insect like aphids, mealybug and scale. This black incrustation is formed which affect the photosynthetic activity of plant. So this is how the affected plant look like with a black incrustation on the affected fruit and affected plant parts. So majorly this sugary secretion or honeydew secretion which is caused by mango scale insect, mango mealybug and mango plant hopper. Coming to mode of spread, the disease leaves serve as a primary source of inoculum. Some scale insect like aphids like Lecanium hemispericum, Lecanium viridae, Lecanium nigrum and other insects sucking pest, they will majorly spread the disease from one plant to another plant. Coming to management, controlling the plant hopper by application of carbaryl or pasmomidone. Spray dilute solution of the starch or maida 5%. We can prepare this maida 5% by boiling 1 kg of maida or starch in a 5 liter of water, and we need to cool and dilute to 20 liter. Then we can go for application, which dries up and the entire the black incrustation come up in a flakes and removing along with the sooty mold growth. So these are some management practice to control sooty mold disease in mango. Then red rust, this red rust which is caused by alga disease, which is Cephalourus fluorescens. Coming to economic importance, it is been observed in Bihar, Karnataka and UP. It first appears in the orchards from 1956 in India. Coming to the major symptom, red rust caused by alga causes reduction in the photosynthetic activity and severe defoliation. The disease can easily recognized by rusty red spot mainly on the leaves and sometimes on the petiole and bark of young twigs and in a epiphytic nature. The spots are greenish gray in color and velvety in texture. Later they will turn to reddish brown and the circular slightly elevated spots sometimes which will merge together completely look like irregular in shape. Then disease development. The disease is common in close plantation. High humidity favors the development of fruiting bodies. The pathogen reproductive and survives in the spots on the leaves or stem and the fallen fruit and any infected plant parts. Frequent rain, warm weather are highly favorable for the pathogen. For worst or poor plant nutrition, poor soil drainage and stagnant air predisposing factor to infection by the algae. Coming to management, avoid close plantation, spray Borax mixture 1 to 2% or Blightax or lime sulfur which can actively control the mango red rust infection. Coming to some other minor disease in mango, first one is scab and stem end rot in mango. which scab is caused by Elisina mangifera, which incidence up to 12 to 13%. For management, frequent application of copper chloride, which will protect the plant from infection by the scab pathogen. So these are the important fungal, bacterial, and some algal disease in mango and their integrated management through chemical, biological, and other important practices. Thank you. And finally, here I am attaching one document related to IPM package of practice for mango for producing quality fruits for export, which is released by Government of India, Ministry of Agriculture and Farmers Welfare. In this IPM package of practice for mango, which majorly covers different integrated pest and disease management practice to cover important pest and disease in mango. They mainly given all the content starting from the planting till the harvest, information related to cultivars, propagation on planting material, crop management, and different types of cultivars available to different ecological regions of India. Then finally, they concentrated on different pest and disease of mango, starting with some important pests like mango hopper and mango mealybug and mango fruit fly, mango bark eating caterpillar. This mango fruit fly very important disease, Bacterial dorsalis, affecting the mango fruit during storage and harvest. And also they given information regarding different types of important disease, what we have covered till now, and their integrated disease management practice to control the disease and pest in a IDM best way.